Hi everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. If you've been following any of my videos on YouTube, you know who I am. You know that my videos are really designed for students who are enrolled in my anatomy and physiology courses at Del Mar College. Anyone out there in YouTube land that finds these videos helpful, by all means, please use them. Anything I can do to help advance any student's life is always a good thing. I wouldn't be where I'm at if it wasn't for some really good professors and helpful people. Now, your instructors or professors may go over some different structures. Y'all may not even go over development at all in your human A&P class. I know we didn't when I was a student. You had to take embryology. But nonetheless, our classes kind of do this because we're trying to prepare nurses for nursing school, as well as many other health science courses. But this is really directed at those who are going to be dealing with a lot with labor and delivery. We're going to be going over development. We've already gone over the male and the female reproductive systems. And a couple of things I want to kind of clarify or go over and point out for my students are these things. By the way, if you find my videos helpful, please hit like, drop me a message, hit subscribe. I'm going to be updating all my videos in the near future. Some of them are very poor quality, but they were made under duress when we went to the COVID break. And so now I have time in my life to try to get them back to up to speed. I'm sorry if I'm cast in a shadow and I keep squinting because of the way that the projector is, but it's the best I got with the lighting and the equipment that I have for now. My goal is to educate my students and not make a great produced video. I'm doing the best I can, so please bear with me. Nonetheless, this shows you an ovary model. We've already gone over the ovary model. We see the follicles here, the primary follicles with an oocyte in the middle, secondary follicles, a tertiary or Graafian follicle that is ovulating an egg. I want to start here because at ovulation, we have the oocyte, this little yellow cell here, and it is surrounded by a ring of cells called the corona radiata. And that's kind of where we're going to start in a minute. And when ovulation occurs, it's going to take us to our next model that I want to show you. So I have a series, two of these right here. I'm going to start with this side. And so what you see here is the vagina, the cervix of the uterus. The narrowing of the uterus is often referred to as the isthmus. We have the body and the fundus. This would be the fallopian tubes. These are the fimbriae. Here's the ovary with the ovarian ligament. This would be called the suspensory ligament, and it doesn't really stop here. It goes to the side wall of the body. This model is a little bit deceiving in that. On this side, you see which ovary has ovulated. And let me make sure you can see this on my screen because I do have a hot spot. Yeah, you can kind of see it. So I'm going to move the camera just a hair so that you guys can follow along here. And unfortunately, that hot spot, that really bright spot on the screen, kind of moves when I move the camera. So please forgive me for making you dizzy, but I'm trying to get rid of the hot spot and still let you see the model that I'm trying to outline here or highlight here. So this is the fallopian tube. Even at this magnification, these sperm are not to scale. I mean, they would still be microscopic, and so would the oocyte. But this is an ovulated egg or oocyte. And one of the things we see here is the infundibulum, or the bend in the fallopian tube. It's about the first 1/3 of the fallopian tube where the oocyte must be fertilized. It has to be penetrated by a spermatozoan. Now, the reason that happens is because as the oocyte is fertilized, it will go from a single cell to 2 cells. And I don't know if you can really see that. I'm going to do my best. Those 2 cells will divide into 4 cells. 4 becomes 8, and then 16, and then 32. And at one point, we have a little ball of cells here where all of the cells are identical. And if I can cut this ball of cells open, it would be a 3-dimensional ball of cells. Almost like having an empty basketball filled with marbles. Okay? And all of these cells in here would have the nuclei and all the organelles. At one point of development, all of these cells are functioning identically. They're expressing the same proteins. Nothing is different from one cell to the next. For example, if I pick a cell here, this cell that I put a little X on, whatever genes are activated and functioning in this cell are the exact same genes that are activated and functioning in all the other cells. They're just kind of these generic cells. We call them embryonic stem cells because every tissue in the body will stem from this original group of cells. Now, At some point, if I have a cell Y and a cell Z here, they are all genetically identical. They have the exact same genes turned on. Whatever genes are turned on or off in this cell are the same ones turned on or off in every other cell. At some point in development, one group of cells will differentiate or become different from the rest. And then all of these cells would start to turn on maybe a couple of new genes and turn off some of the old genes that were functioning. And now they're expressing a different group of proteins. Some of the same ones as these, but a certain group of proteins that might be different. These cells will function differently than the other cells in the other two groups. And if I divide this into 3 groups, then I would have 3 distinct populations of cells. These cells have certain genes on and off. These cells have different genes on and off, and so on and so forth. At that point, we call this differentiation, where cells become different from each other or express different genes and therefore are functioning differently. One of these groups of cells is going to become what we call the chorion. The chorion is what's going to implant or attach to the endometrium here. So this is an implanted embryo. At this stage, we're still at a solid mass of cells, and we call this a morula. It's a solid mass of cells. Morula really means mulberry in Latin, and it's like a little solid ball of cells. At some point, these cells, as they differentiate, the morula will actually start to have these groups of cells start to hollow out and leave an empty space inside of here. And the cells will move apart and continue to divide, but we'll have an empty space in here. When the ball hollows out, then it is called a blastula. And the space inside would be called the blastocele. Oops. And it looks like coele, C-O-E-L-E, but this term means an empty space or hollow space. And in the old Latin, if you wanted to make the letter C sound like an S, there was a letter that had an O and an E together. Modern typewriters couldn't do that, so they still spelled it like C-O-E-L-E. And nowadays, sometimes they're getting rid of the O in there and just making it more Americanized. But nonetheless, this is the stage that we're at right here. It is attached, it is implanted, we should say. The embryo is one group of cells, and then we have what's called the cellular trophoblast and the syncytial trophoblast. You're going to see these terms in just a moment. So, when you see this term sync, to sync things up means that they are joined together, and syncytial and syncytium means a fusion. And so the trophoblast is going to be the blastocele or the blastula, but the syncytial trophoblast is that portion of the blastula that is going to attach here. It's going to become the chorion. The cellular trophoblast will end up becoming the blastodisc will end up becoming the embryo, and we'll talk about that in just a few minutes. So right now, this is all just female anatomy, but one of the things I wanted to show you this model for is because after ovulation, once the egg if it gets fertilized, must go through several levels of development for several days before it can implant. If the egg were fertilized here, by the time it's gone through all these cell divisions, a female has reached menses and will menstruate a fertilized egg, and it would be lost. So we need to fertilize the egg in the first 1/3 or so of the fallopian tube before it can develop enough to become this blastula and implant. So now we're going to use a lot of this terminology in just a minute. I'm going to erase it, and then now we're going to go to the series of the developmental models. And this is really my goal of getting to for lab. We've gone over the breast model in the past. These are the first 2 cells or models in a series of 13 models that we're going to go over. So if you are in my class, we have a series of 13 models on our list of things to know. One of the good things about that list of things to know is when we get to model 1 and go to model 2 and 3 and 4, whatever the number is on the model corresponds to the numbered structures on the list of things to know. I'm going to go over this in just a second. So let me reset my camera because it's just me. I don't really have professional equipment, okay? So I'm gonna go, that's about as good as we're gonna get it. So I apologize for this. Give me just a second. I'm trying to get rid of that hot spot that's on the screen, that real bright spot, because it's hard to see. All right, I hope that didn't make you too dizzy and you can follow along here. I just can't get rid of that hot spot. So for model 1, when the egg is ovulated, this is the oocyte. The oocyte has what's called the female pronucleus. Pro meaning before. There's a membrane that is kind of milky white and almost see-through. It's called the zona pellucida. And then there's a ring of cells that surrounds the egg called the corona radiata. And they come from the follicle when the egg was ovulated. All of the fluid inside the cell would be the cytoplasm of the oocyte, and it would have your mitochondria and all your organelles. Almost all the organelles come from the female, from the egg. Now this little blue dot is supposed to have a little flagellum, but the strings always break off. That's the spermatozoon or sperm cell, and it's number 5 on the list of things to know. So for this model, you need to know the corona radiata, the zona pellucida. Pel means almost and lucid means clear, so you can almost see through it. The cytoplasm of the oocyte, the nucleus of the oocyte, and the spermatozoon. Now once the spermatozoon starts to penetrate the corona radiata, there's a structure at the tip of the sperm cell called the acrosome, or the acrosomal cap. It is filled with several acids and enzymes like hyaluronic acid or hyaluronidase. And these enzymes will actually bore a hole through the zona pellucida, and the sperm will start to penetrate just a little bit and release the male pronucleus, which is the male half of the what's going to become the nucleus. They're called a pronucleus because it's not a nucleus until they're completely fused together. So that's the second model. Now what happens in model 2, though, is once the sperm penetrates the zona pellucida, the corona radiata becomes shed and lost. And then what was the zona pellucida actually separates out into 2 membranes. The outer membrane, number 1, is still the zona pellucida. This area here, all the way around, is a little space called the perivitelline space. And I'm going to write that word out. Peri means around, and vital means life, and lean means little. So the perivitelline space is the little space, or it's the space around the little life thing. So, people can debate till they're blue in the face when conception or when life begins. Is it at conception? Is it whatever? I'm not going to get into that debate. I have my own beliefs and views, but who knows who's right and what science says, because apparently these days science, how true science is depends on what political party you're associated with and which political candidate espouses it, and I'm not going to get into that junk. But nonetheless, the zona pellucida separates out, and this becomes the perivitelline space. This little white membrane all the way around would eventually become what we call the cell membrane, but at this point, it's called the fertilization membrane, because it must be penetrated for fertilization to occur. So here you see the head of the spermatozoon. You see the fertilization membrane, the perivitelline space, and zona pellucida. This is the cytoplasm of the oocyte. Number 2, by the way, is supposed to be pointing to that membrane. And then this is the female pronucleus. Now, one of the things we will go over in some of the lecture stuff is meiosis. The somatic cells, all the cells of your body, undergo cell division called mitosis, except for the gametes. The gametes would be the oocyte if you're female, or the spermatozoon if you're male. Now, during gametogenesis, particularly spermatogenesis, the sperm undergoes certain types of meiotic divisions, from a spermatid to a primary spermatocytes to secondary spermatocytes to 4 spermatids, and then all eventually become fully functional spermatozoa. The oocyte itself starts to undergo the same meiotic divisions. And remember, there's meiosis 1 and meiosis 2. The oocyte becomes trapped in meiosis 1 and does not progress through that unless the sperm penetrates the fertilization membrane. Once the sperm penetrates the fertilization membrane, there are some chemical reactions that happen in here that pick up where meiosis 1 stopped, and it continues meiosis 1. So we see the chromosomes starting to condense and go through the phases of meiosis 1. What you need to know about model 1, corona radiata, zona pellucida, the cytoplasm of the oocyte, the female pronucleus, and the spermatozoon. On model 2, you need to know the zona pellucida, perivitelline space and fertilization membrane, the spermatozoon, or male pronucleus at some point, the female pronucleus with the chromosomes, and the cytoplasm of the oocyte. Now I'm going to go on to the next two models, or maybe it's one. Yeah, it's the next two. So this is model 3 and model 4. And let me make sure they show up well on my screen. When we get to model 3 and 4, really nothing changes very much at all except for the development of the pronuclei. Okay? So, I just did a lap, sorry. I was checking my camera, make sure you could see, and I was trying to get my steps in for the day. No, just kidding. So this is the zona pellucida still. And it somewhat hardens so that another sperm could not penetrate, which would be called polyspermy and usually ends up in a non-viable embryo. Nonetheless, this is the zona pellucida, the perivitelline space, and the fertilization membrane. We see that the female pronucleus is finished meiosis 1, where we would have two sets of chromosomes. In the spermatozoa, those would both divide into another level of spermatocytes. But because we can't have more eggs here, this one's already being fertilized, and this is the male pronucleus, the extra DNA gets packaged in a little bubble of membrane called a polar body. And since this is the first one to appear, it's called the first polar body. And then you see the chromosomes and the centrioles of the oocyte undergoing the second round of meiosis, or meiosis 2. So, zona pellucida, perivitelline space, fertilization membrane. The cytoplasm, this is the male pronucleus. The female pronucleus would be here undergoing meiosis. You see the chromosomes and the centrioles and a polar body. Now, after the second round of meiosis, I now have the male pronucleus and the female pronucleus, the zona pellucida, the perivitelline space, and fertilization membrane. None of that has changed. Only at the second round of meiosis, I still had another bunch of chromosomes I have to get rid of, and so they can be spit out as another polar body. So we have the first and second polar bodies on our model. Otherwise, nothing else changes. Now, the next model, what I would like for my students to know is that for models 5 through 10, I want you to know the stage of development, which is, what does this model represent? This stage of development is called amphimixis. Amphi means a little bit of both, and mixis means to mix. So what's going to happen is we're going to mix the two sets of chromosomes, the 23 chromosomes from dad, or the sperm donor, and 23 chromosomes from the mom, or the egg donor. This is the zona pellucida, the perivitelline space, and the fertilization membrane. I still have two polar bodies. I have the female and the male pronucleus, and I have centrioles here that are ready to separate the chromosomes. As soon as these two nuclei fuse, they're no longer called a pronucleus, they just become the nucleus of the first cell, which is called a zygote. And then that will undergo mitosis and mitotic divisions for a long time. Model 6 is a one-celled human being, although some people will say that's not a human yet, whatever. Whatever you want to argue or believe, that's fine by me. We can differ in our opinions on certain things. But nonetheless, this is the zona pellucida, this is the perivitelline space, and that's a polar body. This is a complete cell. I wish they had sliced it open, but we're seeing it from the outside. And the one-celled individual is called a zygote. So this stage of development is called a zygote. This is a single cell with 46 chromosomes and all the organelles that we learned at the beginning of part 1 functioning to keep this cell alive. Within about 24 hours, this cell is going to start undergoing rapid mitosis, and it will double in number every division from 1 to 2, 2 to 4, 4 to 8, 16, 32, 64, 128, 256, 512, 1,024, 2,048, 4,096, and blah, 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 and become you. Anyway, Now, once we get past the zygote stage, this stage of development is called first cleavage. To cleave means to separate, like a meat cleaver, or breast cleavage or butt cleavage is where things are separated. So this is called the first cleavage. This is called a cleavage furrow. A furrow is like a little, like when you're, hoeing the ground, you make a little furrow. So this is a cleavage furrow. We have two cells now that are called blastomeres. I still have a zona pellucida, perivitelline space, and polar bodies, but now we're at the first cleavage, and I have two blastomeres. That's what the cells are called for a while. Then those two cells will divide into 4 cells, so this is called second cleavage. Zona pellucida, perivitelline space, polar body, blastomeres. Now, this will continue on and on and on. At some point, we get to the stage that is called a morula. And I wish we had taken 2 pictures of this. If you tilted the model over, you would see a whole bunch of little cells going around in a little ring here, and there would be a second layer of cells, bunches of them. We're somewhere around the 16 to 32 cell stage, but nonetheless, a solid mass of cells is called a morula. This is still the zona pellucida, still the perivitelline space and polar body, and then this is still the blastomeres, and you see the nucleus and the centrioles. It's undergoing, oh, that's a polar body there. The cells are undergoing rapid mitosis, and eventually they will hollow out and make a hollow ball of cells called a blastula. The morula stage is here. I believe that's model 10. Once the cells start to reach the blastula stage, they're going to implant. And let me lower this a little bit, because if I get too high up there, you can't see. This is going to be the embryo. This is called the syncytial trophoblast. This is where it fuses to the endometrium. If we were looking at a giant uterus like this, or not a giant one, but if we were looking at the uterus like this with the vagina, and we had the blood lining, which would be the endometrium in here, as a thick blood layer, what you are looking at is this dark stuff would be attaching to, and then we have a little ring of cells. All of this is called the cellular trophoblast. This is the syncytial trophoblast. Within the cellular trophoblast, we have 2 layers of cells here that are differentiating. So it kind of looks like this. The little empty space inside here is going to become the amniotic cavity. This area out here is going to be outside the embryo. It's going to be called the blastocele, or the extraembryonic coelom. This space. Extraembryonic means outside the embryo. And so this space here is called the blastocele, or the extraembryonic coelom. All of this structure here that has cells is the cellular trophoblast. This is the syncytial trophoblast. And we can even see some maternal arteries and veins or blood vessels starting to develop. So this is endometrium. This is the blood layer. This is the syncytial trophoblast attaching. This is the cellular trophoblast. This will be the embryo, and there's 2 layers of cells. This upper pink layer is called the epiblast, epi meaning uppermost, and then this is called the hypoblast, this yellow layer of cells. The amniotic cavity, the extraembryonic coelom, this space here, and this whole thing would be the cellular trophoblast. Okay? So hopefully you got all that. Now, this is the next level of development. And so this uterine wall is still here. These are the maternal arteries and veins. The syncytial trophoblast morphs into a thing that is now called the chorion, and it kind of completely encapsulates the developing embryo in here. This is still that extraembryonic coelom or blastocele. Okay? Now, what happens at one point of development is there's a little bit of a sinus here, little gaps that develop between the chorion and the endometrium, and blood will leak into here, and so those empty spaces filled with blood are called maternal blood sinuses. It's also at this stage of development where sometimes a female can have a little bit of spotting, and if she knows she's pregnant and sees some spotting, sometimes they go, oh, maybe I'm not pregnant, if they didn't want the pregnancy at that stage. Or some people who want their pregnancy get scared. Oh, I'm bleeding, is something wrong? Very often it's when this stage is happening, a little bit of blood can leak out. So check with your physician, always be safe, but nonetheless, that's the maternal blood sinus. This is called the chorion, and it will develop these little finger-like extensions which increase the surface area for the absorption of all the nutrients and gases, oxygen, and the exchange of carbon dioxide out. Now, those are called chorionic villi, the little finger-like structures. What we called the epiblast in the last slide has now become this ring of cells, which is going to be called what we call the ectoderm. So there's going to be 3 germ layers, 3 layers of cells from which all your tissues will develop. The ectoderm, ecto means outside and derm means skin. So the ectoderm is going to be your skin, and actually, oddly enough, it will invaginate and form the neural tube in your nervous system and some of your endocrine cells. They're all derived from the epidermis. This is going to be called the endoderm, but right now it's called the yolks. Actually, it's called the ectoderm and the endoderm. The endoderm is going to form some of the inside of the body, and it's also going to develop part of what we call the yolk sac. There's a third layer, this green layer, that's eventually going to migrate right across in between these two and become the middle layer. Meso means middle, so that is the mesoderm. So we have ectoderm, endoderm, and mesoderm here in the green. They start to hollow out, and that's going to be part of our neural tube and form a lot of our body. So nonetheless, this would be the extra embryonic coelom or blastocele out here. So here's what you need to know if you're in my class. Endometrium, maternal arteries and veins, the maternal blood sinus. The brown layer is the chorion with the chorionic villi. The pink cells are the ectoderm. The yellow cells are the endoderm from the epi and hypoblast. And some other cells are going to migrate across here. This green cells right here would be called the mesoderm. This space would be the extra embryonic coelom or blastocele. Hope you got all that. Now, eventually the model goes from looking like a jellyfish into looking somewhat like a tadpole. What you don't see here is this is the endometrium. This would be the uterine cavity. These are called the chorionic villi. And when we magnify them, we can see they have developed blood vessels. These are embryonic blood vessels developing from an embryonic heart. This is the heart. This would be called the head fold. That ectoderm is here in pink. The mesoderm is this green layer down the middle. And the endoderm is going to be this yellow layer, and it's going to actually become part of our insides. Now, the heart develops what will eventually become the aorta and the inferior vena cava and some blood vessels. And some of those blood vessels are going to go through this area here. This is called the body stalk. Eventually, it will become the umbilical cord, but at different stages of development, it's called different things. That's what makes embryology hard. Or difficult, is that at one stage it's called the syncytial trophoblast, then it becomes the chorion, then it becomes the placenta. And the body stalk will become the umbilical cord eventually. So these are the embryonic blood vessels growing through here so that as the maternal blood sinuses leach nutrients and oxygen in here, it can be carried into and pumped around, feeding the cells for this rapid growth and development. And any toxins can come out and go back into mom and get filtered out at the kidneys and the liver. So here's what you need to know. Heart, head fold. This is the mesoderm. This is the yolk sac starting to develop, the body stalk, and the chorionic villi. The next level of development, we look even more like a tadpole. We look kind of like an empty worm or a hollow worm in the previous one. This is sort of the tadpole stage. Now, this over here would all be endometrium. This would be the uterine cavity. And we can see that some of the cells of the uterine cavity have kind of encapsulated the entire developing embryo. These are the chorionic villi, the maternal blood sinus that we saw before. This is now called the yolk. This is the body stalk, which becomes the umbilical cord. Here we can see the head with the folds that will become part of the brain and a developing eyeball. These are called vestigial gills. The term vestige means something that's left over from before. And so the term vestigial, which will mean leftovers, and those are called vestigial gills or gill slits. So at one point of development, we have gill slits here. This is the heart. We even have a long tail. These little block strike-like structures can be called somites. They're going to develop into vertebrae and some of the ribs. But we actually have a long tail, and eventually it will evolve away during development. And then this is going to be the amniotic membrane, the white, or the amniotic cavity. So, chorionic villi, body stalk, the yolk, the heart, we can see the embryo, and the amnion. Now, we're almost done. We have a couple of models left to do. So now, this gets to the pregnant uterus models, which we've already covered in the previous video. And so that previous model would be here, but now it's starting to look distinctly human after the first month, or closer to human. And then we have the decidua basalis, the decidua capsularis, and all the other structures. And then eventually, by month 2, we look very distinctly human. We're about the size of a human thumb, and our entire body is there. You see arms and all the structures. We've gone through these models in another video. So I hope that was helpful and informative to you. I hope that helped you understand a little bit about how development goes throughout pregnancy and why some of the structures fuse and what they become, so that you have some kind of clue about what's happening in the human body and the female body during pregnancy. If I were you, make sure you read the chapter, look at all these terms. It's up to the student to take the terminology we throw at you, what's in the textbook and what's in the literature, and put it all together into a bigger picture. We can't do it all for you. So get to work, start learning this stuff. I hope you had as much fun as I did. I hope you learned something. I hope this helped you learn the development models, and I hope to see you in another video. Thanks for watching.